This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 71, recorded on January 16th, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth, and our first podcast for 2014. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello. Happy New Year.
You too. You have a good New Year so far? 16 days?
You know, I'm still on my feet.
Can't ask for more. Also joining us today from the Medical University of South Carolina, Michael Schmidt.
And happy belated birthday, Vincent.
Thank you, Michael. Always nice to have a birthday right after New Year.
Yeah. Well, at least it's not at Christmas time. It's just right after Christmas.
It's fine. I don't need anything. I don't actually need to get older, but it's inevitable.
This is true.
It's
Time
okay.
and tide waits for no one.
My mother used to tell me she figured I was going to be a New Year's baby, but it didn't happen. I guess that's fine.
So did you save them some money in the tax year, or did you arrive before the first?
No, January 2nd.
Second.
Oh, okay.
No, I didn't. January 2nd.
Pokey.
Well, you know, my younger son was due on December 26th, and I thought he would actually be born on my birthday. I made a bet, but I lost. He was born on January 6th, 4 days later.
A mind of his own.
Close, right?
Yes.
All right, on this first TWiM of 2014, the first of many, we have two papers which have been published recently, both of which have to do with the gut microbiome. And I purposely did pick the two in the same area. And the first one is an MBio paper recently published, which is entitled "The Gut Microbiome Modulates Colon Tumorigenesis." And Michael is going to tell us all about this one.
Well, this paper from a group at Michigan and Baylor College of Medicine in Houston, Texas, is really, I think, a primer on how to do cause and effect for looking at the role that the microbiome will play in disease processes. They're really this group that is led by Pat Schloss at Michigan, and the bioinformatics and sequencing was done by Joe Petrosino's group at Baylor. They really, this paper really teaches us how to look at a very complicated story in a very straightforward and clear manner. So I recommend this paper if you're trying to get into the microbiome, because it's really very readable. You don't get overwhelmed with lots of color figures showing us, all these different bacterial levels going up and down. They really tell an elegant story. So I'm going to give you what Elio likes to say is the sum. And so we all know that there are trillions of bacteria in our gut, and these things are known collectively depending upon which side of the definition you want to look at, whether or not it's the genetic contribution of the bacteria that influence the process or whether the host is involved. I think this story is agnostic as to whether or not you have to believe one definition or the other. But recognize that the gut flora is very important for the normal functioning of the human intestine, as well as the intestine of this animal system that they're looking at, which are mice. The second thing is they bring in from the published literature that evidence is out there that suggests that disruptive changes in our gut flora are strongly correlated with the development of colorectal cancer. That is why all of us over the age of 50 have a shiver run up and down our backs every time we see a hose the colonoscopy. So every time we see a garden hose, the shiver runs up and down everyone over the age of 50.
Michael, they say the risk factors are eating meat, alcohol consumption, and inflammation. So this alcohol consumption part, is it okay if I have a glass of wine every day?
As long as it's red.
Okay.
As long as it's red. And so
like hard liquor, tequila and scotch, that's bad, right?
Well, that leads
especially before breakfast.
Well, no. I mean at night.
Well, it leads to fibrosis of the liver and all sorts of other consequences with the liver. And the liver is really in charge of what's going on in our gut, along with the pancreas. So those two principal organs really, I think, can have a dramatic influence on not only who's in our gut, but how many of the who are there. And that's the other really neat thing this group did, is they not only defined who's there, but actually how many were there. And that will become very important as you go through the story with us today, to not only ask yourself the question, who's there, but how many are there? They bring up, or they use a particular statistical methodology that allows them to compare and contrast not only who, but how many. And so the principal question that they addressed is pretty straightforward. And it starts off with, how does the gut microbiome change during the development of this very horrible cancer, colorectal cancer? And they accomplish this task by using a tremendously efficient mouse model of inflammation-driven colon cancer. And their mouse model is very elegant. It uses, your standard mouse strain, C57 black, and the way they generate tumors in these animals is they expose them to a carcinogen. The carcinogen is then followed up by an inflammatory substance, dextran sulfate, and they administered the dextran sulfate three times. And then within a very short period of time, literally within 70 days of the administration of this carcinogen, you have frank tumors in the animal. So you can well imagine what they have done is they give this carcinogen at day 0. They use fecal samples from day probably -7.5. So they collect samples before they give the carcinogenic agent. And then they subsequently collect fecal samples from the animals that they expose to the carcinogen and the subsequent dextran sulfate on days 20, 21, and then they expose again to DSS, and they again collect stool samples. And they did this only using 12 animals, and they ask a very simple and straightforward question, who's there? And they did something really clever. And this is all clearly laid out in a very simple set of figures in MBio. The first figure in panel A, we see the capitulation of the generation of tumors and when they do things, when they collect their fecal samples. So it's real easy to follow along. And then they simply ask the question whether or not the animal is developing tumors after the introduction of the carcinogen and after each round of the dextran sulfate. And what they find is that tumors do indeed develop by day 73. And so then they ask the next logical question is, well, does the microbiome actually contribute to this process? So any limitedly trained microbiologist would say, well, let's deplete the flora. And the manner in which they depleted the flora was, again, pretty straightforward. They effectively added 3 antibiotics to the animal's water. They added metronidazole, vancomycin, and streptomycin. They gave that to the animal 2 weeks prior to the carcinogen, the AOM. And then they continued to have the antibiotic in the water. And they asked a very straightforward question about tumor penetrance and tumor incidence, which effectively gives you, do tumors develop and what do the tumors look like? And the antibiotic-treated animals had significantly fewer tumors in the colon than the untreated mice, and that had a great p-value of 0.001. And more importantly, in those animals that received antibiotics that developed tumors, the tumors in those animals were much smaller. So I always am thinking as I'm reading these papers and asking myself these questions, and it's one of the things I'm going to torture my GI colleagues with is, so should human individuals who get a positive polyp exposure, should we think about giving them a course of antibiotics to effectively trim things out of our guts if we've developed polyps? Or should we think about transitioning to giving them an anti-inflammatory, because colorectal cancer is known to be facilitated by inflammation and inflammatory insults in our gut. And so whether or not we may want to think about anti-inflammatories and a course of antibiotics, especially if you have an indication for growing polyps in your gut. And again,
Michael, have any studies been done using anti-inflammatories or aspirin, for example? Antibiotics?
I don't know. And I started to do a lit search when I was doing this, when I started reading this paper on Monday, but then I got interrupted by the beginning of the semester. So.
Excuse me, I can comment on that. So in addition to the microbiologist and Joe Petrosino, who's the expert sequencer, they needed to recruit a physician for this project, and that was Grace Chen. She's on the faculty here in the Division of Hematology and Oncology, and many of the experiments were actually conducted in her group because she has expertise in looking at the development of cancerous tissue. You'll see from this manuscript that there are just some beautiful pictures of the tumors and the normal tissue, and that was really thanks to her expertise. But I was able to communicate with Grace, and we're getting a little ahead of ourselves, but she said definitely their results make them wonder if they can shift the microbiome of patients over to a healthy microbiome to reduce colon cancer risk. And then something we've talked about before, we're getting kind of excited about the idea of doing fecal transplants.
Oh, yes.
And this story raises the important issue that we've got to be careful what the composition of that fecal population is, because we don't want to predispose somebody inadvertently to colon cancer.
This is the icky part of the ending. So we have
Right.
to put the public disclosure. The end of this story will be a little icky if you... And Michele already let out the icky part, the fecal transplant. And that's actually one of the cool aspects of their papers, how they actually recapitulate the linkage of the microbiome. And so as we begin to tease through this data, keep that in the back of your mind as how you will actually, it's sort of kind of like Koch's postulates with a twist. And because the microbiome is now a community rather than being an individualized cell, and they were really able to do it nicely. And so after they got that great antibiotic result, they then asked whether it was the specific populations within the microbiome that were essential for tumors. But they know from the literature again that there's no real clear-cut microbe implicated in the development of colorectal cancer. There have been previous studies in the literature. So then they began to ask questions about numbers. So to determine whether the relative change in the bacterial density following antibiotic treatment was due to the change in the bacterial load, they performed quantitative PCR. And again, their sentinel molecule was the 16S ribosomal RNA gene, because the 16S ribosomal RNA gene is actually a reflection of whether microbes are growing, and you can actually quantitate based on the amount of ribosomal RNA as opposed to DNA that you see as to whether or not you can actually physically then count the players that are present. And what they found is that the number of 16S ribosomal RNA gene copies per milligram of fecal material was not significantly different from the antibiotic-treated samples to those from the untreated stool samples. So again, it's not so much the number of the bugs. And again, there are microbes that are resistant to the 3 antibiotics that they use to treat, and consequently, this then led them to posit or conclude and then ask, the results indicated that changes to the structure of the community rather than the total bacterial numbers were likely responsible for tumorigenesis. So then what they did is they began to assess the community that was present, and they did this in the traditional way using the similarity indexes. And what they found is that there were significant shifts in the microbiome were associated with this colon tumorigenesis. And that takes us to the second family of figures. And their color scheme is they go blue, green, and red, as well as black. Black is before they expose the animal model to the carcinogen. The blue is the fecal samples associated with the first DSS exposure. The green is the second DSS exposure, and the red is the third DSS exposure. And what they showed is that the mice showed a significant decrease in microbial diversity in their gut microbiome following the first round of DSS administration. And what DSS in concert with the carcinogen are doing is DSS is effectively providing the inflammatory component to their model. So the inflammation is actually influencing the community, and that you can actually witness in the black bars versus the colored bars, and they're statistically significant. And so they then did this next fancy statistical trick, which is called the ordinations of distances between fecal samples. And what they found is at the time of when they sacrificed the animals to actually look at the tumors, do the pathology and whatnot, is the tumor-bearing mice developed a significantly altered microbiome that clustered very differently or separately from the baseline samples that were taken prior to the first round of that inflammatory dextran sulfate. So that led me to really focus in on figure 2C, which is this it looks like a glorified dot blot from a Petri plate or a colony count. And what they did is they did non-metric multidimensional scaling, which is they abbreviate NMDS ordination, based on a theta YC distance calculation for the 10 mice that they had in their carcinogen inflammatory model. And this particular technique, or this theta YC, as I framed it in my head, most similarity indices that are out there only consider the likelihood of who's there when you're comparing two populations. They don't really consider how many of them are there. And that's what this particular paper, it's actually reference 42. So just write that down and actually hunt up reference 42. And it's reference 42 that really gives you an inkling of why the method they used is so remarkable in helping them form their conclusions. They not only know whether the two populations of the tumor model and the control are different, but they know how many or their proportion are there. So this similarity value, this theta YC, actually takes both the type of microbe in this population that is present and the volume or the number. And if you think about it, the intestine is this processing device that eats stuff. And we all know, because we've all seen the ads on TV about eating more fiber and, eating less meat, and all of those things influence the flora. But now here in a model that they can actually recapitulate the development of colon cancer in 73 days, they're actually showing the importance of the microbiome in that process of developing cancer. And it's through non-culture dependent techniques. They're just basically isolating stool and sequencing it. And then because they can run it through this theta YC algorithm that enables them to figure out what's going on, they're then able to demonstrate number as well as who's there. So it's really a very powerful methodology to know what's actually going on.
So it's especially compelling because they were able to do a time series. They first collected samples before treatment, and then as the inflammation and tumors developed, so they could watch this shift in the type of species and the number of the species with time. I think that made it really compelling.
And that's panel D. And I was so excited with panel 2C. And then what Michele just said is actually panel 2D. So those two figures really are the gotcha data. This is where they get you. So now any Stanier trained microbiologists out there, the ones that understand selection and substrates influence or enrichment technology that we all learned in our second course in microbiology, will now appreciate this next series of experiments where they now are going to demonstrate it's the bacteria that are actually facilitating the development of the cancer in concert with the carcinogen and the inflammatory process.
And in simple terms, they need to address cause and effect.
Mm-hmm.
So far we have a correlation, a beautiful correlation in figure 2, but then the really beautiful work is next, yeah?
Yeah.
So that's the question. Did the tumor cause the bacteria to change or vice versa?
Right.
Yes. So how do you imagine, this is the icky part. So if you don't like icky, fast forward on the tape to the next paper. But this the icky part. So what they did is they took germ-free animals. So these are animals that are born truly sterile and free of microbes. They then grow them up to a particular point in their lives, and then they are said to conventionalize them.
Where does that word come from?
That's the icky part.
Is that a disguise for a fecal transplant?
It's actually from the gnotobiotic community, and gnotobiotics predate fecal transplantation.
I see.
But it simply means, as Vincent said, is they're doing a fecal transplant, and the way they do that is they provide the formerly germ-free animals stool from the animals that had the cancer and the inflammatory substance given to them. And so they get fresh stool. as well as the bedding. So this is the dander that's shed off of the animal and the stuff that comes sloughing off as the stool goes through. So you're going to get dead skin cells and all sorts of other things in the fecal content and the bedding. And so they just literally dropped the germ-free animals into two separate cages, one with healthy animal stool and bedding and one with stool from the animals that had tumors. And they continued to re-inoculate the animals with transferred fresh feces and fresh beddings to these two groups of germ-free animals, which there were 10 in each group.
And these mice get the AOM DSS, right? Or do they not? They do?
No, they don't. No, they don't.
They just get to live in the home of mice that did get that.
Correct.
Plus and they had tumors. I guess they had antibiotics.
Well, I believe they still had to treat them.
Yeah.
With the carcinogen.
Oh no, you're right. I'm sorry. Following conventionalization, the mice were treated with AOM DSS, and as they had described it previously in the first figure, where they measure and then they witness the change. And the conclusion they drew from this very straightforward experiment is the animals that got the tumor flora, if you will, actually developed more and bigger tumors than the animals that got the healthy flora. So what this is showing us is, as Vincent alluded to in the beginning, is that it may be possible to do stool transplants to affect either the prevention of colon cancer, and this could be very important if, we know that colon cancer is familial. And so if you have colon cancer in your family, you may be able to look at the opportunity to get the right probiotic that is considered healthy. But, we should caution everyone that mice are not people. And the C57 mice are all genetically equivalent.
Yeah.
And, people we're basically a mongrel species. Everybody is different. These C57 black mice are all pretty much genetically very, very equivalent.
Hey, Michael, this experiment, it caused a twofold increase in tumors in the mice living in the house of the tumor-bearing mice, right? Mm-hmm. Now, when you give mice antibiotics, the increase in tumor burden is much greater than that. So why is it only twofold when you give the fecal transplant, essentially? I didn't
figure that out, and I was hoping Michele pimped her former colleagues to see what their speculation on that was.
Well, the antibiotic treatment actually reduced incidence, correct?
Yes, it did reduce incidence, but I think what Vincent's asking...
Yeah, so I'm sorry. My question is, if you just give mice the...
antibiotics.
The treatment... No, not antibiotics. I meant the AOM DSS. Okay, you get 20 to 30 times the number of tumors in untreated mice.
And is that in a germ-free, or that's...
Well, it's pathogen-free. It's not germ-free.
Okay.
Mm-hmm. So
they have normal flora. They have normal flora.
Yeah, so is in the germ-free, when you put them in the cage inhabited previously by tumor-bearing animals, you get a twofold increase. And you're also giving them the AOM DSS. So I'm just curious as to why...
the healthy flora is overwhelming the bad flora, because remember, mice will process the stool that's in their cage. They'll actually eat it. So you're effectively displacing normal flora with the stool.
Right, but it's only a twofold increase in tumor burden, right?
Yeah.
I'm just wondering why it isn't more. If you're giving them the
healthy stuff,
the tumor flora, as you put it, should be more, I guess.
Well, the answer may be in the inflammatory component of the model, the DSS. The DSS just may result in sufficient inflammation to overwhelm the good behaviors of the normal flora. And they allude to that when you begin to look at their last figure in their manuscript, which is figure 4, and you look at, again, that green and black dot type axis where they're looking at the model values of what's actually going on, looking at the theta YC distances with a healthy microbiome or the microbiome of the dysbiotic community. And that may actually then reflect what's actually going on, the community dynamic.
So these germ-free mice pick up the tumor microbiome, essentially. They did look at that, right?
Right. Yes, they did. They did indeed look at that. So,
and actually, that was another analysis that they did. Is they not only, they cataloged who was there, and so they can begin to correlate particular species with lack of tumors and other species with prevalence of tumors.
Right.
So the hope is that ultimately we can identify particular species, or more likely particular metabolic pathways that are protective or inflammatory, and then develop a really powerful or effective probiotic rather than fecal transplants.
Right.
And they talk about that. I was going to this is the last aspect I was going to discuss in this paper is the real microbiology, is the shift in the relative numbers or relative abundances and actually numbers of the bacterial populations. And they look at the gut flora, and the anaerobes are actually the big players. And they talk about the Bacteroides, members of the Bacteroides family being very important, and the Porphyromonas, as well as the Erysipelathrixae. And so again, they're effectively looking at these microbes that are thought to convey good health, and then they transition into a narrative in their discussion about inflammation being central to the condition that fosters the genotoxic, if you will, because in order for cancer to commence, you need a trigger. So they have the carcinogen in concert with the constant perturbation by the dextran sulfate that will then facilitate the production of the genotoxic reactive oxygen species, as well as the pro-tumorigenic inflammatory mediators, TNF alpha, IL-6, IL-1 beta, and interferon gamma, and of course IL-23. And so it's really about the number of the microbes that are there, because the number, again, think gene dosage when you think of the number of microbes, because it's about turning over substrate. And here's where they introduce us to our second paper, where they begin to talk about butyrate and the importance of butyrate in modulating good gut health. And of course, butyrate is a fermentation end product from many anaerobes, but it's also a tremendous carbon source that you just can't perfuse butyrate into the gut, because other microbes will actually eat it. And so,
although they've done tests with butyrate containing enemas, I think, right?
Oh yes, but the way they finish off their paper is they talk about, or they hypothesize that bacterial populations that serve a protective role in our important mediators of gut health, at least in the murine or mouse microbiome, is the Porphyromonas family, that will actually facilitate the production of these short-chain fatty acids, of which butyrate is probably the biggest player. And so it's these short-chain fatty acids that are thought to convey good gut health in the mouse, and we know that this also holds in the human as well. And so that is a really neat story. To sum it up, the first thing is they have a really outstanding model that develops a colon tumor within 73 days. They then sample stool over the course of the 73-day period. They'd ask not only who's there, but how many are there. They show by the addition of antibiotics that the microbes are important, because you get more tumors in the presence of microbes when the carcinogen and immune modulators are the dextran sulfate is present. And then they recapitulate their model by bringing the bedding and the fecal material from healthy and diseased animals or dysbiotic animals back into a germ-free thing. They effectively conventionalize them, and they show that, yes, indeed, if you add healthy flora to even animals that have been exposed to inflammatory conditions and a carcinogen, you can actually have fewer tumors. So it's an elegant piece of work that is eminently readable.
So one of my confusion points here is that in people, as far as we know from the studies that have been done, you can't say any particular community profile of gut flora is associated with colorectal cancer, right? And that, as you say, may be because in part we're outbred. But in these mice, there's a consistent pattern of certain species going up and others going down. So how can we get around that if we want to treat people somehow with a probiotic? If we can't identify what you need, how do we deal with that?
It's the next step. The alpha analysis is who's there. The beta analysis that they talk about is not only who's there, but how many are there. And then the next form of analysis, or the tertiary, is I think we need to ask the question, what is the relative abundance of particular sentinel enzymes?
Right.
And it's the sentinel enzymes.
Metabolic enzymes, most likely.
Mm-hmm.
And so that crosses us into the threshold of our good friend Joe Handelsman's area, and that's metatranscriptomics.
And metabolomics, I would argue, right?
Yeah, the metabolomics is the next step, and we're going to need that quantum leap in looking at, akin to what Malditoff has done for the clinical microbiology world, of figuring out who's in a pure colony. They can identify the individual organism, but we're going to now need that quantum leap in Malditoff to effectively assess the metabolomic profile of a healthy colon and a semi-healthy colon, a diseased colon, and then a cancer colon. And there's probably a continuation, and I'll bet you that there's someone out there trying to develop the proteomic techniques using this very model of colon cancer because it's so predictable in space and time.
And if I could put that concept in simpler terms, it's if you imagine you're designing a garden, you might want something tall and red in the back and maybe something short and yellow out front. And there are lots of different species that can give you that, but you want those features tall and a particular color.
Nice.
So if we're focusing too much on the species, it might be distracting. If once we learn what the metabolic profiles are that those species bring, that might be more informative and give us a better handle on the way forward.
That's great. Yeah, it's perfect.
And using the garden metaphor, we also have to consider when the flowers bloom.
Right. And how they interact with one another.
Yeah, it's very similar to pollination, where you need the bees to pollinate the fruit of a particular flower. And I think a lot of it has to do with succession of populations that go up and down. And we cannot divorce ourselves from thinking about the contribution that the host makes in terms of modulating the population by dumping in our hormones and when we eat and when we defecate, and all of those things are probably very much involved.
Yeah, I wanted to mention that because they make this point that an inflammatory environment in the gut is probably promoting and amplifying progression to colorectal cancer.
Correct.
So if you're genetically predisposed to making IL-23 that's a little bit more active than someone else, that could be a factor, right? So all of these...
Well, we know the P53 story for colon cancer in humans, if you have a lesion there.
Sure, there's P53. There are the colorectal genes that predispose, but there are going to be many other host determinants that play into inflammation. And it may sound seemingly impossible, but if you focus on the products, as you guys have said, the metabolic products, if you can figure out which ones are doing this, you don't have to worry about the genetic part. You don't have to worry about the microbiome. You just treat the condition in the colon.
Right.
Right.
So my challenge to the really clever computer programmers out there is take Michele's garden metaphor and use various different colors of flowers over time, put a temporal component into it as well in order to present some of these data.
Right. And it may be in humans, which, as Michael pointed out, we're all very different. If instead of looking at the species, once we get to the level of metabolic pathways, maybe then a pattern will emerge.
Yeah, exactly.
All the noise will fall out, and we'll see what is in common.
Yep.
But before we move on, I'd love to emphasize what an interdisciplinary team Pat Schloss put together to make this project work. So the first author is Joe Zacular. He's a PhD student, and he first met Pat when he was an undergrad at UMass Amherst. I was able to talk with him, and he really gave a ton of credit to the Honors College at UMass Amherst, which works really hard to get their students thinking about experimental biology early on in their career. So he met Pat in an honors course his freshman year that had maybe 5 or 10 students in it. And then UMass Amherst had a program where the students could then do internships. So Joe was able to go to Wyeth in Cambridge, a pharmaceutical company at the time in Cambridge, and did an internship, came back and was really excited about science, ended up moving with Pat to Michigan, where Pat, who himself was trained as an engineer, as a postdoc with Joe Handelsman, started to learn microbial ecology, was actually working in a beetle model at Amherst, but by coming here to the medical school, he could collaborate with physicians like Grace Chen and now ask these questions in context of a human and human disease. So Joe was, in addition to working with another PhD student, Neil Baxter, and then a master's level bioinformatics specialist, Catherine Iverson, in the lab, was feeling just so fortunate to be able to have not only Pat, who's got the really strong computational expertise, but also working really closely with Grace Chen and her colleagues over in internal medicine, where he learned a lot about the human side of this problem. So Joe's advice to the listeners out there is to really get a strong foundation in a number of disciplines and follow that where your interests go. So again, microbiology is a big tent. We'd love to recruit engineers, mathematicians, computational biologists, chemists. There are a lot of great questions to ask.
Even graphic designers.
Yeah.
I understand he's a golfer.
And we've golfed. Yeah, Joe's a pleasure to golf with.
It's like the old days bankers used to golf. Now it's the microbiologists. So we've done now this paper, which is colon cancer and the microbiome. We've talked about obesity in mouse models and the link to the microbiome here on TWiM. We have talked about asthma and in mice and the link to the microbiome. So many different human diseases associated with changes, and maybe they're going to be mechanistically similar in that it comes down to metabolic products. Right? And it's not just worrying about the particular bacteria and the genes, but what's made. So I want to take a few points from the discussion to move into the second paper, and that's about butyrate, a short-chain fatty acid, butyrate, which Michael mentioned. And as he said, this is a product of the fermentation of fiber, soluble fiber in the gut by bacteria. And in this paper, they point out that this compound butyrate reduces inflammation, and that's the link to fiber, or one of the links to fiber. Having a high fiber diet and having reduced risk for colon cancer is that perhaps the butyrate that is made reduces inflammation, which, as we've talked about, could be a problem for pushing cancer development forward. And there are links between having high fiber and decreased colorectal cancer risk. They say that people who have low fiber diets or have colorectal cancer make less short chain fatty acids, including butyrate. So all these links between butyrate and inflammation and colon cancer. So that is a little lead into the second paper, which was published in PNAS just recently, and it's called The Microbial Metabolite Butyrate Regulates Intestinal Macrophage Function via Histone Deacetylase Inhibition. So again, butyrate is this short-chain fatty acid. It is, as we said, has some link to diets high in fiber and reduced risk for colorectal cancer. There have been some studies done where they give people butyrate actually to see what happens. The point of this paper is not colorectal cancer, but rather how our great immune system allows a microbiome to be there in the first place. There obviously is a conflict. You have to have a great immune system that keeps pathogens out, but it has to be able to allow this very beneficial microbiome to flourish. Okay, and so they're focusing on soluble molecules because we know from many other studies that soluble molecules are how bacteria can communicate with each other, and they know that butyrate and propionate, which is another short-chain fatty acid, are common in the gut. So they decided to look in the gut and see if there would be an effect of butyrate on immune cell function. So in other words, could it be that butyrate, we know butyrate is an inhibitor of inflammation. Could it be that its other role is to keep our defenses down so that the beneficial bacteria can stay there? So that is the thesis of the paper. So it's different, but really linked still to the first paper.
And whose work is this, Vincent?
This is from Yale University and the Max Planck Institute in Germany. The authors are Pamela Chang, Li-Ming Hao, Stefan Offermans, and Ruslan Medzitov. He's a very well-known innate immunologist. Yes, a contemporary. Well, he was trained by Janeway, who was a critical person in really establishing our understanding of the innate immune system.
A real thought leader.
Amazing, and he's no longer with us, but Ruslan is carrying on his tradition. Really, a fantastic scientist, done all sorts of cool things, and
also at Yale.
Yeah. Okay, so butyrate we've talked a little bit about. A couple of things which might be useful while we're discussing this. Butyrate is known to be an inhibitor of histone deacetylase. Histones control the expression of DNA into mRNA. Whether they're acetylated or not controls that. When there's an acetyl group on them, it's thought that they're more actively, transcriptionally active. When the acetyl groups are taken off by deacetylases, the transcriptional activity decreases. And what is thought to happen is that butyrate binds to the active site of the histone deacetylase and inhibits it. So they look at what's going on with butyrate and with immune cells. So the first experiment they do is simply to take macrophages from bone marrow, and they put them in culture. They stimulate them with lipopolysaccharides so that they will make cytokines. And then they either treat them with butyrate or another short-chain fatty acid, propionic acid. And then they measure inflammatory molecules. They measure nitric oxide, IL-6, IL-12P40. And what they find is that, in fact, butyrate decreases the production of these inflammatory mediators by these bone marrow-derived macrophages, okay? They do measure a few other cytokines, TNF-alpha, the chemokine MCP1. Their levels are not affected. So whatever the effect of butyrate is on inflammatory mediators is rather selective.
And a really good place to look at as you're going through the many panels of flow data on the cytokines, plus and minus the LPS and the TSA, is to tear out figure 5D and just keep that in the back of your mind as you're reading through these things, because it's a really simple cartoon showing you how butyrate gets into the system, how it acts on the macrophage. The net consequences, it results in the HDAC inhibition, which then concomitantly reduce the nitric oxide levels coming out of the macrophage, the IL-6 coming out of the macrophage, and the IL-12, which are the pro-inflammatory cytokines. So that is how the butyrate is acting to lower the inflammatory response.
Yeah, so that figure is a good to keep in mind when we're explaining this. Okay, so those were bone marrow-derived macrophages. Next, they look in the colon. There are macrophages in the lamina propria, which is this kind of loosely assembled connective tissue below the epithelial layer. And there are a lot of macrophages in there because they are patrolling and seeing if there are any pathogens coming in from the gut lumen. So they isolate these colonic lamina propria macrophages. Again, this is from mice. And they, again, treat them with LPS, with or without butyrate. And in fact, they show that they have the same effect. The butyrate reduces secretion of IL-6, nitric oxide, and so forth. All right. And next, they look at feeding mice butyrate. Can you feed mice butyrate, and what does it do? Again, they do this, then they isolate the colonic macrophages, and once again, the levels of cytokines go down. This time they measure the RNA production by quantitative PCR, and there's a substantial reduction in their production in the presence of butyrate. Okay, so...
And they used our old friend from the last paper, dextran sulfate.
Yeah.
To effectively cause inflammation, and they administered the DSS in concert with the N-butyrate.
Right. So that's seeing if butyrate can overcome that inflammation, right? Mm-hmm. Okay, so they've established that macrophages, either from the bone marrow or the lamina propria in the colon, can respond to butyrate and make fewer or lower levels of the certain cytokines. They do a few experiments to try and figure out how butyrate is signaling, how it's working, what kind of pathway is involved. And I don't want to get into these because they're both negative. It doesn't involve toll-like receptor signaling, and it doesn't seem to involve G-protein coupled receptors, okay?
And it should be pointed out that the senior author of this paper is effectively the inventor of toll receptors along with Janeway.
Yeah.
And they needed to do those experiments because there was a precedent in the literature showing that butyrate can, in some circumstances, alter TLR signaling and G-protein coupled receptors. Right. So they needed to address that. But as you said, negative results.
Negative results. Okay, so how does this work? They go to the knowledge which we mentioned at the beginning, that butyrate is known to be inhibitor of histone deacetylases. So they have a positive control, a compound called trichostatin A, which is an inhibitor of the HDACs, and they can measure histone acetylation. They have an assay for that, which they can do by Western blot. And in fact, the positive control trichostatin A does a nice job of inhibiting deacetylation of the histones. And they also find that butyrate basically inhibits the enzyme because you see more acetylation of the histone in a dose-dependent manner when you add the butyrate. So that's by Western blot. So you have an antibody to the modification, the lysine that's being modified, and you can measure that directly. They also do another experiment, which is pretty cool, which is called CHIP, chromatin immunoprecipitation. And in this experiment, you take chromatin from cells, and you add an antibody, in this case to the lysine on the histone that's being modified by the acetylation. And then after you add the antibody, you break up the DNA, you immunoprecipitate, you pull out the chromatin that's bound to the antibody, and then you say what DNA is associated with this histone. You do that by PCR. Okay, so you can look exactly at what transcriptional regions are being modified at the histone level. So when they do that, they see, in fact, that they see an increase in acetylation of histone lysine number 9 at the promoter regions of the nitric oxide synthase gene, IL-6 gene, IL-12B, etc., but not TNF-alpha, and that kind of correlates with the results that we saw in the cultured macrophages before. Okay, so 2 lines of evidence showing that butyrate is inhibiting HDACs. If modification of histones is being affected, you would expect to see an effect on the presence of RNA polymerase at these promoter regions. So they actually look at that. They have an assay for looking at RNA polymerase 2 at promoter regions. Of course, when RNA polymerase 2 goes to a promoter, that means that it's going to transcribe there or would like to transcribe. And what they find is that treatment with butyrate or TSA, trichostatin A, causes a decrease in the amount of polymerase, RNA polymerase 2 bound to these cytokine promoters again. So again, there's this inhibitor of HDAC is reducing transcription at these specific sites. Now, it's actually the opposite of what you would expect, because leaving acetyl groups on histones should make it more active, but they have an explanation for that. They feel that the acetylation is recruiting a repressor complex to these genes. So it's kind of the opposite of what you would predict. In fact, it tied us up for a bit when we were talking about that. So that's what's going on there. They try and understand how butyrate is taken into cells, and they look at a particular transporter called the monocarboxylate transporter, but that doesn't seem to be involved in the uptake of butyrate. And the last experiment is to take a mouse model of colitis, and again, they use dextran sulfate administration to mice, and it gives them acute colitis. And then we know in that system the inflammation is due to infiltration of macrophages in the innate immune system. So the question is, if you give these mice butyrate, will it ameliorate their inflammation? So you give them DSS with or without butyrate in the drinking water, and you look at them for 7 days, and you can weigh them as a readout of colitis, and you can also do histological examination. But unfortunately, there was no effect of butyrate in this system.
It's probably because the bugs in the gut are eating the butyrate that the mouse is eating through its water.
Yeah, that could be. And that would overcome any effect of butyrate, right?
Yeah, you'd almost have to gavage them from the back end in order to and really load it up. But it's a concentration thing, is my suspicion. And remember, all of these fermentation end products that are produced by the anaerobes in the gut often have, one microbe's waste product is another microbe's food source. So these things are often, it's not only the issue of are you making the compound, but we're going to have to not only figure out when the compounds are being made, we're also going to have to figure out where the compounds are being made. So then thinking about the microbiome, you're going to have to figure out where the microbes are spatially in the colon, because I would postulate that the butyrate, the most valuable butyrate producing microbes are the ones that are firmly attached to the epithelial layer, so that the butyrate is actually driven to the epithelial layer so that it could then interact with the macrophages that are in the lamina propria. And it's probably an issue of where the butyrate is in the gut. So the mere act of just physically giving the animal the butyrate in its drinking water may not deliver a sufficient concentration of butyrate to the epithelial cell layer that is excluding the microbial population from the lamina propria. So it's likely the microbes that are on that are attached to that epithelial layer that are so important. And we know that to be the case for some instances from other microbiome papers. But if I was going to go and harvest the microbiome, I would actually look at who's attached to that tissue.
Yeah. I think that's a good possibility to explain this. So I thought that, they have shown in this paper that, in fact, butyrate can inhibit cytokine production by macrophages, but that may only be part of the whole story.
Mm-hmm. Right. In fact, there's another group, a Japanese group of Ono, who published in a Nature paper in 2013 that butyrate can induce T regulatory cells, which are in general immunosuppressive. And in their model of colitis, they found that butyrate actually did dampen colitis in that mouse model through a Treg pathway. So I think there's still hope for butyrate and treating the inflammation in the gut that can predispose to colitis.
Yeah, in fact, there have been 2 papers recently showing that butyrate's involved in the production of these regulatory T cells in the gut. As you mentioned, there's another one showing that they're involved in the periphery too. But I think that this is a reductionist approach, looking at just the macrophage. So that may be part of it. So the idea is that butyrate is produced by microbes, the good microbes in our gut, our flora, that is telling the macrophages, we're here, don't trigger inflammation. And that's what keeps our gut flora able to be there. But my question is, what happens when a pathogen comes in and then the macrophages have to secrete inflammatory cytokines? How do you differentiate? What's the signal? And that, of course, we have no idea. So lots of questions still coming out of that. By the way, inhibitors of HDACs are currently in clinical trials for a variety of neurological, inflammatory, and infectious diseases. So,
stay tuned, as the hosts say.
This has already been figured out that, regulating transcription might be a way to treat certain kinds of diseases where there are lots of genes being turned on that you want to be turned off. So there you go. Butyrate. That's the common theme. Let's answer a couple of emails here. We have some neat ones. A whole bunch have accumulated since last year. And the first one comes from Zachary, who wrote a very simple email. How long does it take to become a microbiologist?
I don't consider myself done yet.
I have a feeling Zachary is like a high school or an elementary student, who's doing a project. And,
yes.
So Michele is, you guys are the real microbiologists.
Well, I would tell Zachary and other people like him, what I did is I was a biology major in college, but I didn't do any research at all. But if you have a bachelor's degree in a science, you can get a position in a laboratory as a technician. So they'll bring you in at a kind of a base level. You start contributing in a very basic way, and then you'll rise to the level at which your skills take you. So it's a great way to sample research science, see if you like it, and then you can commit to getting more education in the area that you're most interested in.
But I think you could also, according to that, you have to go at least to college, right? But maybe...
Well, there are medical technology programs that you can get through, and you just have to go through the regulatory. They often end up with a baccalaureate type degree.
Right. So, but I would guess that even at a high school, you could get a job in a microbiology lab and work in it and become reasonably accomplished. And you can't go very far, of course. You might want to go back to school, but it depends what you mean by becoming a microbiologist. You can certainly work with bacteria and contribute with a high school education, I would think. But as Michele said, you'd probably want to do more than that and at least go to college.
But high schoolers who are fortunate to be around a university or a hospital could possibly do some volunteer work in the summer.
Right.
And get some exposure and advice from the professionals there.
And then, of course, if you're like Michele and Michael and myself, you took probably 10 to 12 years to become microbiologists. And as Michael said, we're not even there yet, right?
No, it's a lifelong adventure.
Well, we're microbiologists, but we just don't know the answers to everything. We're learning.
That's correct.
We're still learning every day. Okay.
I'll say one more thing to Zachary and his cohort. We're fortunate in this country that there's very good funding for graduate school in the sciences. So our students don't pay tuition. It's covered on grants, and they get a stipend. So you don't have to build up a bank account before you start graduate school.
All right. Yeah, I think volunteering is always a great way just to see a field and
get advice from professionals.
A high school student, a local high school student, came to my house last night because he knew I exist, and he said, I want to volunteer in your lab and learn how to do science. I'm thinking of being a scientist, So, you hear about someone who's near you who might do something you like, and just try it. I said, well, I'll try and help you. All right, the next one's from Tim. I've never heard of magnetotactic bacteria before and thought perhaps you guys might find this interesting. Not sure if you've mentioned these on TWiM before. He sent us a review on magnetotactic bacteria. And Tim is our friend in Minnesota who is a farmer and writes to us often. And Tim, we did talk about it on TWiM number 25, magnetotactic bacteria. And Michael might remember that one.
I do. When I was a first-year graduate student, it was actually one of the projects that was being worked on in the lab is could they cultivate magnetotactic in the laboratory using traditional culture techniques? The short answer to that long story is no. But now folks have finally figured it out, and that's now literally some 30-plus years later.
So, Tim, go check out 25. We had a good discussion. Elio was there, and he actually had done some things with it as well. Benjamin writes, I listened to your podcast that discussed the primary literature article, a Burkholderia pseudomalate toxin inhibits helicase activity of translation factor EIF4A, that was published in Science. The article mentions that after the crystal structure for BPSL 1549 was determined, and they saw that it was a similar structure to CNF1C, they created a mutated version that was not toxic, which they expected, since the same happens in the mutated E. coli version of the protein. Does this mean that if they find an agent to inhibit the protein, that it would likely be able to work on both E. coli and B. pseudomalate toxins, since they are similar? And is this a normal technique for treating bacterial infections, since by targeting a protein rather than the bacterium, it would leave the bacterium alive to make more of the protein toxin?
Those are big and good questions. The answers will be loaded. It depends on how your inhibitors function and how specific the inhibitor is. While the crystal structure generally gives you an idea, you really have to test the agent in order to find out. And oftentimes it's the toxin that actually does the dirty deed. And fortunately for us, vaccines are a much better way of inactivating toxins because you use the full force of our immune system to do it rather than giving an ethical drug to an individual that would take it out. This way, the individual has if you will, the drug already on board if they've been vaccinated, while if you have a toxin producer and you don't get to the physician or you're unable to fill the prescription, all of the things, the toxin may do you in.
Are there any current drugs or antimicrobials that target toxins? I'm not aware of any.
I'm not either.
Okay.
And just to be clear, Benjamin, when we are vaccinated against toxins, your body makes an antibody that acts as a direct inhibitor of that toxin. So it's the same principle as you bring up, and it works beautifully.
So his point is that if you target the toxin, the bacterium would still be able to reproduce, right?
That's
so
right.
when we are immunized against, say, diphtheria, which is you're immunized against the toxin, right?
Right.
Correct.
Why doesn't the bacteria? How does that work? Why are we not worried about the bacteria multiplying?
Because your immune system's taking out the toxin. There are many people that are indeed colonized with C. diphtheriae, which is why we continue to vaccinate the population.
So we don't worry if the C. diff is still there, and it's just the toxin that we're worried about.
It's a purely toxin-mediated disease.
And I guess are there cases where the toxin is essential for bacterial growth?
Not to my knowledge,
No?
but I'm sure there's always an exception to the rule.
Okay. All right. So I would say also to Ben that even if two proteins are structurally similar, that I think they're pretty lucky in this case that the mutation had the same effect in both proteins. I would say that's not always going to happen because things are determined by often very small differences. And so I think they were lucky in this case. This paper, by the way, we discussed on TWiM number 30. The next one's from Ludwig. Hello, TWiM team. I recently saw an NPR article on the disease SCID-X1, an X chromosome-linked immunodeficiency disorder where the body has no functioning immune system. I was wondering if I could hear your thoughts on how the microbiota would respond to this. I know prior research has shown that the immune system plays a role in shaping the community composition of the gut flora. How would the microbiota react to a complete absence of an immune system from birth? Thank you, and keep up the great work.
That's a question for our notobiotic friends out there. Have SCID mice been placed into notobiotic colonies? I'll have to ask my colleague here who runs our notobiotic facility if she's ever done that. Michele, do you know?
I don't know.
I mean, there are plenty of people out there with immunodeficiencies, right? So what happens to their microbiota? Do they go wild? Do they overrun? Is it not an issue?
Yeah, that particular mutation won't take out the whole immune system.
Yeah.
So it's a little bit more complex than that.
SCID-X1. I'm not familiar with.
Well, in 1993, my colleague Ed Baelish published a paper, Mucosal and Disseminated Candidiasis in Notobiotic SCID Mice.
Yeah.
And Ed, unfortunately, just recently passed away in October from cancer, and he was one of the pioneers. Ed, for many years, was at the University of Wisconsin at Madison, and when he retired the first time, he failed retirement, so he and his wife relocated to Charleston because they no longer wanted to shovel snow, and he worked here on and off over the last probably 12 years since they retired from Madison. And so Ed actually published on that, and I'll actually give Vincent the link to this paper that just came up when I looked, because I knew Ed was a great pioneer in mucosal and disseminated candidiasis, and he always used notobiotic animals.
Trying to see here. SCID, normally in SCID, just plain old X-linked SCID, you lack, or you have very few T cells and NK cells, and you have B cells, but they don't work well because they don't have T cell help, and you get lots of infections from a very early age. But I'm not aware if anyone's looked at microbiota. It's a good question, right?
Yeah.
Mm-hmm.
I have been recently seeing papers on the idea that, in fact, when we are infants, part of the reasons why our immune system is deficient is to allow the microbiome to establish, which then, of course, is believed to play a role in helping our immune system to develop.
And the gut tissue to develop as well.
Yeah. All right, let's take one more. This is from Will, who writes, Hello, TWiM team. I'm a senior biology major who is bent on pursuing a career in microbial ecology. Last summer, I discovered your podcast, and I wish I had started listening earlier. You guys have been so great that you inspired me to start a biology talk show at my university's radio station entitled Disentangling the Bank, for the iconic concluding paragraph of Charles Darwin's On the Origin of Species. It's a 1-hour program where two co-hosts and I summarize papers we found interesting that week and talk about the major results and implications. because of your thorough preparation, I'm always telling them to go to the primary source, much to their chagrin. we can't go as in-depth as your show, not only because we lack the knowledge which comes with being in the field for years, but also because this show is aired live to the public. This results in us heavily summarizing each paper. Nevertheless, we have a great time discussing the articles off-air in depth, and it has become a sort of running joke that I always have a microbiology paper each week, never taken from your show, though. Keep up the great work, and thanks for the inspiration. You could take papers from our show. We wouldn't mind.
Yeah, or suggest some for us, Will.
I think that's a great idea to do a talk show. You should do that with your Michigan radio station, Michele.
Oh, I think TWiM keeps us busy and TWiV keeps us busy enough. Thank you.
I know you have science to do. I have always thought of getting the Columbia radio station to air our recordings whenever they have some dead time, at 3 a.m.
Mm-hmm.
And that's pretty cool that you can do it live. If you had been listening today to us live, you would have heard a lot of stuff that doesn't end up in the final.
A lot of head scratching.
In the final episode. So I admire you doing it live.
He's very brave.
I did find his radio show on Facebook. They have a Facebook fan page, Disentangling the Bank. So I'll put a link to that so people can find it. I don't know where you are, but I guess we can't get your broadcast. I'd love to hear it. All right, that's it for TWiM 71, the first of the year. And you can find this recording and all the others at microbeworld.org/TWiM. There is an archive page there where you can get a listing of every episode that we have made to date, including the ones on magnetotactic bacteria and many, many others. You can also find TWiM at iTunes. And if you use iTunes, please go over to the site for the show and give us a rating. You leave some stars or comments. That helps to keep us visible in the Apple ecosystem over there, the iTunes podcast directory. The more people comment on your show, the easier it is to remain visible and be discovered. Do send us your questions and comments to TWiM at twiv.tv. Michele Swanson is at the University of Michigan. Thank you. Thank you, Michele.
Thank you.
You having a good sports season?
We are. We're undefeated in the Big Ten, but we've got a tough stretch coming up.
Do you have season tickets?
I do.
Wow. Were you on the
football and basketball?
Football and basketball. This is really amazing. I had a conversation with Ann Palmenberg, who is at Wisconsin. Mm-hmm. She also has season tickets.
That's who we're playing on Saturday, and they're currently ranked number 3 in the nation, so we're going to have our hands full.
Where are you playing them? Madison?
There. Yeah, at Madison.
It's very noisy in Madison.
Yeah.
Ann will be there. Wow. Well, good luck.
Madison is the one with the raised floor, right?
Nope, that's Minnesota.
Oh, that's Minnesota?
I was in Madison in November, and my hotel was across from the stadium. It was during the week, so there was nothing going on there, but it was very impressive, as I'm sure the stadium at Michigan is.
It's the most impressive for anyone who watched the Winter Classic and saw the Red Wings play the Toronto Maple Leafs in a snowstorm here on January 1.
I never did understand that. I watched that game, and, it was like, I kept saying, do I need to clean my glasses?
Apparently, all the players really loved it. It reminded them of their youth, playing outdoors.
Yeah,
but we digress.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent and Michele.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and in particular Chris Condayan and Ray Ortega for their technical help. The music that you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.